Welcome everybody to the December 11th IMAG MSM working group on multiscale modeling viral pandemics and the Global Alliance for Immune Prediction and Intervention meeting, and our last seminar of the season. And we will hear from Job, which is an awardee of a prize for the best reproducible model. We will see an announcement for the next one for this year. So let's move forward. First of all, the usual people that are involved in this project, the co-lead, Tom Ellicott, James Glazier, administration and activities coordination, myself, James Luca, but also Ellen and Maria, which are online. And if you have any question, anything, you can reach out to any of us. We have a number of channels for communication. We have a Slack, we have our main hub, IMAG MSM wiki page, where all our collection is stored. We have our YouTube channel, and we have LinkedIn and our website of GLIMPRINT, where we will see it's renewed, and we can find also many interesting information down there. And you might have seen this slide already, just to say that we are also creating the Embryo seminar series, and which are connected to GLIMPRINT and the community, which try to put together people from different community interested in multiscale modeling, but also experimentalists, etc. A call, if you have publications to share, and you would like to have it included in our collection, please get in touch with us. Of course, this is the first time I say this today. If you have such a publication and you will even like to come over and talk to us in one of these seminars, please come forward and get in touch with us, and we will be happy to host one of your seminars. Our renew GLIMPRINT website is now on, and we can now post a job announcement, and is also a hub for again for connecting modelers experimentally around the concept of immune prediction and intervention. And please check out all the QR codes and links, but if you miss them, don't be afraid, because everything will be available again at on our different schedule for a coming seminar. Really, we have planned well, this is the last of the season, so we will have a Christmas break until January, where we have starting planning a lesson learned from COVID-19 pandemics, and for the attempt to modeling the origin of this working group. And we are trying to arrange an exciting mini seminar series about that. But then we are also getting applications for, just proposal for having seminars in the new season. And please don't be shy. Suggest yourself, suggest your students, suggest your postdoc or PhD. We are always happy to hear from many different perspectives. And I think, oh no, here we go for the announcement. A few important slides. Announcement. Things that are happening soon, and you want to, perhaps you are interested in applying to those. Multiscale Immune System Modeling Center announces inaugural pilot award request for proposal. If you're interested, letter of intent due December 15, so you need to be fast. Proposal due January 31st. Here QR code and links, and again, they will be up on our usual links. We are announcing our 2026 OpenVT reproducible agent-based multicellular model competition. The first awardee of this competition is our speaker for today. And if you are interested to candidate yourself for the next round, please get in touch and use these links to submit your application. Last but not least, we have Cypher Macmillan Multiscale Human School happening in May, if I'm not mistaken, and you can find everything at this link. If Maria, you would like to say a few words about that, if I'm missing something.
Yeah, sure. No, I actually the deadline to submit is next week, I think December 17th. And if you are successful, CFAR will actually cover all the costs. The school is happening in Vancouver, and we have actually teachers and speakers from both fields, theory and organoid development. And this is all towards building a virtual human embryo.
Fantastic. Thank you very much. Okay. Do we have any other short announcement from our also community? Otherwise, if we don't, without further ado, I will introduce our speaker for today, Job Berkhout from Utrecht Uni, which is going to discuss toward the virtual embryo, especially he will talk about neural tube closure and how these defects are so important on human health, and how we can have computational approaches to try address some of these issues.
All right, so today my talk will be about towards a virtual embryo, and maybe also fitting for the summer school announcement. So first part of the talk will be about the paper which I submitted to the OpenVT competition, which won first place, and then I will talk about some additional additions I did to the model to actually being able to predict chemical disruptions and toxicity using agent-based modeling. So birth defects are very important. They happen 1/3 to every live birth, and as you can see, they are quite severe. So if you look at this image, you see here various let's see a bit later, various babies suffering from such neural tube closure defects. There's open spina bifida where the back is open, or seventhly where the skull is malformed. And they can be caused by chemical exposures. We're exposed to a lot of chemicals during our daily life, such as fumes from factories or certain products in our food or pesticides that are sprayed on our fields. And all of these chemicals can induce sort of a birth defect. And currently, there's no suitable testing strategy available. So we have our animal models, but of course, they are, if you use your rats in the lab, for which you need many, you run into, for example, ethical concerns, or they are very slow, their animal models, and you need way more. We need a way greater chemical coverage to address all these different kind of chemicals we take on in daily lives. They are very expensive. There's a high resource cost. You don't really know what's going on, and also with animals, they are limited predictable for human health. So we need a new solution, and I will tackle the approach of computational modeling, asking the question: can we sort of build a virtual human that we can use to predict whether these chemicals are disruptive or not for human development? And we're not going to build the whole complete virtual human today. We're going to take a case study on the neural tube closure, which is the basis for the spinal cord and the central nervous system. So the neural tube closure is an early event of pregnancy at the end of the 3rd week, and it's a very complicated process. And a lot of different chemicals act on this process, and that's why it's a very important case to discover and to build models of it. They happen quite often, with 3.5 out of every 10,000 cases in the US. And for example, if you have spina bifida, how it would look clinically is that the back is open and the spinal cord protrudes outwards, being exposed to the outside world, which causes very severe handicaps to these children suffering from it. And there's no direct test available for risk assessment. So we're going to build a new test strategy in the form of a computational model. But to model something, you need to understand it first. So first, and I want to tackle this concept, going to sort of go through a blueprint where we're going to map the neural tube closure to properly understand it, and then we're able to build a model from it. So if you're going to build something, we also basically, for a house, we also make a blueprint, or for something like a space station, we also make a blueprint. So I thought, why not for a biological process like neural tube closure, where we map all the things that are going on, what's under the hood of this biological process, to be able to so we become as knowledgeable as possible, so then we can build computational models from it. And we're not going in blind. We have these certain frameworks that guide us, where we have physiological maps or adverse outcome pathways. And physiological maps map the healthy state, and adverse outcome pathways, they can map the disease state. And did this with a student of mine, Alasha, from the slides. So first, what are the physiological map? Well, physiological map defines the healthy neural tube closure, and we map the molecular events that lead to the cellular events. So if we zoom into this place, for example, this process, dose-to-lateral point bending, we see all these genes and proteins that have a role in this process, and this is basically what needs to go correct to be able to get this cellular process, dose-to-lateral point bending, go right. On the other hand, AOPs, adverse outcome pathways, link the molecular disruptions to phenotypes, and they sort of highlight which cellular events are most important. Say, if you remove one of these key players, BMP4, you get a downstream key events, things that stop working, but then can end up in a neural tube defect. So if we look at the processes, for example, successful endpoints formation, we see this. It goes well, and the outcome is correctly. However, if we look at the disrupted state, so BMP4 key player is removed from the process, we then look at the disease state, and we look at a patient or a section, and we can see something like this, where the hinges, the bending points, are not properly formed. And which agent-based modeling we can link these genes and tissues? Because normally, if you look at development, you don't look at boxes and arrows and stuff. You actually look at the formation of specific morphogenetic structures. So for example, these examples here on the right. And with these age-based models, we can visualize the phenotypes, because cells follow rules derived from gene expression, and this collective cell behavior can lead to the tissue level outcomes shown here, where it goes right or it goes wrong. And with Computational, with age-based modeling, we can simulate biology. And I think most of you are familiar with this tool or this process, but on the left here you see the cells. On the right you see how it look in Computational 3D. And we have like an environment in which you place our cells, and cells can move around and act in this environment and exert cellular behavior. And we use this concept to simulate neural tube closure. Neural tube closure is a complex process, as I mentioned earlier, where you need to start from a neural plate all the way down to sort of the closed tube. And we have a number of steps where the neural tube or the neural plate goes through. So you start with the flat ectoderm, you see cell proliferation and formation of a stiff structure in the middle, the floor plate. The neural folds elevate. You see physical forces that push the neural plate inwards. Cells continue to proliferate and excrete certain protein variants, and at the end, the neural plate bends and fuses in the middle. And certain cells, neural cells, migrate away to form other essential structures of the central nervous system. And when we look at this complex process, we can ask ourselves, okay, what's essential? Those are like the key events, which we derive from the AOPs. And how are they regulated? For that, we look into the physiological map. And the most essential key events we determined were the floor plate formation, where the neural plate sticks to this green dot notochord to make a stiff structure. Also a hinge point formation, where the neural folds need to bend in order to fuse, and neural crest cell domination, where neural crest cells move away to form other essential parts of the central nervous system. So by having these key events, we can now extract sort of a gene regulation network from the physiological map we developed earlier. And how I went over this, I went from the back to the front, basically. So first, look at the most essential key events, which are here located in the map. Then get the proteins or genes that directly regulate these key events. Then include the receptors that signal to us these genes and proteins that regulate these events, and include the proteins that attach to these receptors, and at last include the processes that secrete these proteins. And if we put then all this together, we get sort of this gene regulation network that we can place into our agent-based model to be able to mimic a neural tube closure across time. So this is basically the engine powering the model. And how I simulate it is I simulate it very simple with Hill functions. So every positive arrow and activation is like a positive Hill function. Every inhibition is an inhibition Hill function, scaled between 0 and 1. So very Boolean-like, so to say, where everything acts a bit like a switch that can be turned on and off depending on the cellular state of each cell during the simulation. And if we put it together, we get like this Computational 3D simulation of the neural tube closure. This is the cell field. And we see all the behavior we talked about earlier integrated in this spot where you see the formation of the stiff structure. We see growing of the cells. We see bending at the hinge points here over here. And at last, we see fusion at the midline where some cells migrate away to form all the structures of the central nervous system. And if we then place our virtual embryo neural tube model next to a real embryo, we see a lot of, we see it's quite the same. We see here in our virtual embryo, we see the innovation similar to an actual human section of neural tube. We see this, that it becomes steeper and narrower in time. We see also this real embryo, we see the bending of the neural plate, and at last we see the fusion. And because we have this large gene network integrated in our computational model, we can also visualize all these gene expression chains across time. So we see if our cell field, the same simulation as earlier, but now we expressed in each cell the gene genetic state. And we see sort of a patterning going on where we have sonic hedgehog from the bottom, BMP from the top right, log in a bit across the cells, patches also here, and snail acting later on. So we actually see this patterning, what we see in biology happening. But that's the question, how well does it compare to actually biological studies? We can place the model with actual in vitro hybridization data side by side, and you see that it's quite similar actually. You see, for example, sonic hedgehog going down from the bottom to the top. You see patches as well being activated here. PAX3 at the top here, our threshold as well. So we see that we can mimic these patterning quite well in silico when we compare our agent-based modeling to actual in vitro hybridization data. So if we put it all together, we basically make like a dynamic systems model where we have our gene expression, and this gene expression triggers cellular responses, and all these cellular responses trigger tissue morphogenesis, which then can cause a developmental outcome. And now we can use this concept to model neural tube closure defects by adjusting the fold change of key genes in the network to simulate perturbations. If there are any questions, you can just speak up, by the way. I can't see the chat. Let's see. So you can ask yourself, all right, we have this AOP, small AOP, where we have like one gene inhibited, which then causes certain steps towards a neural tube defect. And we want to validate this model against known biological data. So we can try that by, for example, look at the wild type, the healthy situation. What happens? We get a complete neural tube. But what happens if we, for example, remove one of the important players that have an important role in one of the key events? We get a different situation where the model doesn't really close very well. And actually, the phenotype is quite similar to what we saw before, where the dorsolateral hinge points are not able to fold well, and we're not able to get a correctly closed neural tube. Similar to, for example, if you make another knockout, where rather than we get a stiff floor plate down below, we get sort of this ball of cells that is very unstable, which causes fusion in these images. Or we remove another important protein from the network, where we get sort of a similar effect as the being before, but with a slightly different touch, where it sort of fuses the bit line, it's still able to close a little bit. So if you look at these wild types and these different knockouts, we see that the model actually can predict observations in biology quite well. And of course, because it's a model, we can quantify these predictions. So we run it a bunch of times in different scenarios. Here we have knockout scenarios, and here we have hyperactivation scenarios. We see a distinct pattern with all these knockouts and hyperactivations, where we find a different probability of a defect to occur. Where in noggin, we mostly see the malfusion defect, or in BMP4, where it's an evenly spread. But if you, for example, hyperactivate noggin, you get towards a different type of defect, a closed neural tube defect, for example. And these three types of defect are defects you can observe in patients in, for example, hospitals. So where do we go from here? Because now I showed like this, very briefly, this neural tube closure model, where we can use knockout studies to see if it does result in a probabilistic outcome, phenotypic outcome. But what we actually want to do is to be able to actually use these kind of models to toxic or not. And a chemical does not often generate complete knockout of a specific protein. So we can integrate, and also one idea with these kind of models is we have it as a platform where you can integrate different tests, toxicity tests, for example, into these virtual human model that also generates safety profile. So the test can feed into our age-based model, but also directly into the safety profile itself, making NAM or a hazard assessment without the need of animal models. So now the more fun or the new part is actually use this model for actual probabilistic hazard assessment rather than just knockout studies. And basically what you want to do, or what I did, is basically when you do like RNA-seq studies, right? You put like a chemical, in this case we use the zebrafish. You get like this heat maps out there if people work with RNA-seq data. And what we want to know is, all right, we have this RNA-seq data set. What happens if we put this in the model? Like what's the outcome? Will it be fine, or will we get a closed, an open, or a malfusion effect, for example? And so I went to the lab, did zebrafish embryo studies. I don't know if people are familiar with lab work. Highly recommend, tons of fun. All right, it's better. All right, so we use zebrafish as a model system. And zebrafish is quite powerful because it has neural closure dynamics that are conserved with vertebrates, which is very nice. So for example, if you use a cell has not really the mechanisms to be able to perform neural tube closure, for example, but the zebrafish has these kinds of mechanisms conserved. It's all animal model, so it has developmental relevance. The zebrafish also allows morphological assessment, where we can see, all right, we can dose the zebrafish embryo with chemicals and see how does it look, how does the development look, is the head malformed or the tail maybe, and look at the bone structure. And then we do an RNA sequencing analysis to link the molecular response to phenotypic outcomes. And because the zebrafish neural tube develops within 24 hours, doing samples basically means you have to be on the lab Lionel Richie style. So we put different experimental stages of zebrafish sampling. We pick these 5 stages because they are all relevant for specific moments, where the bud stage is very the early onset of neural tube closure, and the 24 hours our post-fertilization moment represents post-neurulation. And these intermediate stages are all critical for specific cellular behavior, where you have the floor plate here, does a transpoint formation here, and neurocrystal elimination at the final stage. And because you need a lot of actually experiments, you need, of course, put on some nice music for the fish so they have a good time. If you compare these stages to a human, you see indeed that the bud stage is the onset, and then all the 3 critical key events are captured across this time. This is how we look under the microscope. You have, for example, these somatite stages, and what you do is basically you count the somites, and then you know which stage they are. For this to align the RNA sequencing data, so all the protein genes are properly aligned. And when you plot this, you can link basically the molecular disruption to phenotypic outcomes, where the neural tube closure mechanisms is a bit of repeat, I think. Yes, so when we align this staging, we can then plot the neural tube, the RNA sequencing across time in the PCA. And what you see is that every time point, every stage, has very distinct RNA sequencing patterning, where at the first stage is completely different than the last stage, really highlighting the complexity of this process, of this neural tube closure process. And also that you have these specific genetic programs during neural tube closure that all represent specific cellular mechanisms. Then we took this assay and we exposed chemicals to the zebrafish. Here are the chemicals used. I will go over a couple of examples, and we try to expose them as early as possible. And for example, if you expose them with cyclopamine, which is something that can disrupt the sonic hedgehog pathway based on our analysis. See, for example, one of the key events, flow plate formation, is negatively regulated. And if you compare at 5 days old, the fish to the vehicle, the healthy situation, you see quite a big difference. Where with cyclopamine exposure, the tail is quite crooked, skull's a bit malformed, thus the vehicle, everything looks fine. So another example, for example, Tiran gives a lot of different up and down regulations in RNA sequencing. And you also see that, and what's most clearly visible is that the spinal cord is completely crooked, completely isometric. Compared to a vehicle, it's more like a corkscrew thing going on that you can see with the GFP positive collagen. And Wind and gap junctions are very disrupted under Tiran exposure. But another example, for example, glyphosate. There's not really a major phenotype. The fish looks a bit smaller compared to the vehicle, but other than that, that doesn't have a severe phenotype going on. And you see here some nodal cord development is downregulated, as well as some extracellular matrix organization. Maybe that's why they're a bit smaller, but no major disruptions. So we then can integrate this RNA-seq data within our computational model. So what I did is I basically isolated all the gene family members that are in our computational model. And we concatenate them, these other individual genes into sort of families using the weighted mean. Because, for example, if you look at BMP4 or PAX, there are like multiple types of PAX or BMP4, right? But we only have like one BMP gene in our age-based model. So we just took all the BMPs and put them together, calculate the weighted mean, and generate a family that we then can feed the model. And in this specific cyclopamine example, This is cyclopamine. You see two gene families that has fairly downregulated, patches and NKX2. And you also see a dose response going on where at the lowest dose, S3, the downregulation is much less severe compared to the middle and highest dose. So I wrote an R script that can do this for all chemicals and doses and whatnot. So, I mean, everything will be on the GitHub at some point. So you can do all these analysis yourself with my data or with your own data to generate these weighted mean plots of all your exposure scenarios. Or if you want to look at other things, maybe those scenarios work as well, as long as it is RNA sequencing data. And lots of things are a bit around the midline, as you can see. I mean, cyclopamine has two very lowly expressed genes. Glyphosate is not as much going on. Tebuconazole has more highly expressed genes and low expressed genes as well. And basically, with these RNA-seq patterns, we can feed this to our age-based model to see if we can predict potential toxicity. So how do we actually feed this data to the model? Well, we have this gene regulation network, right? And we can apply a multiplier to every node in this regulation network. I apply a multiplier using this calculation. So if we have a minus 1.5 log fold change, it's a multiplier of 0.35. So then we do this to our case or 3 case studies. And what we see is we have our control situation, which looks fine in these circumstances. And we have our cyclopamine exposure, but where actually the neural tube is not closed and it's kind of crooked to one side. You also have like glyphosate exposure, which actually not much is going on, as we saw in the fish as well. And TRAM exposure, where the neural tube is completely open, completely messed up. And you also see a big bend going on at the bottom, so it becomes very asymmetric, something that we see in our fish experiments as well. Just like our knockout studies, we can then quantify these predictions with, and these are the main defects. So here we have all our chemical exposures. We did all the analysis of the RNA-seq data, and we integrate everything in our computational model. Again, it's the SAM what's shown here on the error bar, and we calculate the probability of a neural tube defect, where a neural tube defect will occur. And what we see is, for example, in the highest dose, is, for example, that TRAM has almost 100% cases of open neural tube closure defect that you can see here. Cyclopamine also has a lot of different varied neural tube defects, imidacloprid as well. Glyphosate is quite fine compared to the rest. Caffeine, a little bit more. And lower doses are still some defects going on as well, more intermediate. And at the smallest doses, only in tebuconazole, there are really a lot of defects going on still. We can plot this. For example, we can plot any defects, aggregate them together. What you see is actually some sort of dose response going on in most cases, where you see, for example, cyclopamine, first highest dose, we have a lot of high probability of defect. A middle dose is still an average probability, and the lowest dose, the probability of defects actually gone. Same for TRAM, where you have two very high probabilities and then a very low probability in lowest dose, similar to the control, for example. Tebuconazole's toxic in all cases, and in imidacloprid, you see a very clear dose response going on. What's interesting is that with something we did not see during our knockout studies, is that actually we see different types of defects going on with different chemical exposures. So here on the right, for example, what we often saw, what you also saw in the fish, is that the neural tube becomes asymmetric. We quantify it as asymmetry. And for example, in TRAM, we often saw the neural tube becoming very asymmetric in the highest and middle dose. What we also saw is that the skin, the blue part, goes inside the tube, mainly with tebuconazole, that prevents the closure. We also saw very often is stenosis, where the neural tube is closed, but the gap is very, very small, which causes developmental problems across time. Another issue, for example, is neural cell delamination inhibition in orange, where the neural cells are not able to delaminate properly, so parts of the central nervous system are not formed correctly. And at last, gap in the ectoderm, where the surface is not completely fused, but the neural tube itself is closed. And you can also, if you look at all these subtypes, for example, tableconazole, where we did not really see a big difference in total neural tube defects, we do see across those that the amount of subtypes becomes smaller. So at the highest dose, the neural tube defects that we saw were much more severe compared to lower doses. So also here, there is still dose response going on. I think that's around the end. To summarize and talk a bit, we mapped the zebrafish neurulation RNA-seq to gene families in our age-based model. We are able to drive simulations with transcriptomics, where we saw the chemical-specific fold change in modulate or silico age-based model. Our age-based model was able to predict types of morphogenesis and neural tube defect types. The prediction is aligned with the zebrafish morphology, and we can basically use this as an integrative mechanistic tool where we can integrate different types of data within a single platform to be able to perform probabilistic hazard assessment of the chemical toxicity on neural tube closure. So this, of course, is one small step, but I hope a giant leap for animal-free risk assessment. And with these age-based models, we can protect human health and the environment. And as we no longer need animals, maybe they can go to models for us. Of course, it was not all me. I had a lot of help from my promotion team and my external advisors. So my promotion team, Harm Heusingveld, Al Piefma, and Juliette Lechler. And so advisors, James Glacier, Tom Knudsen, Richard Spencer, and Giulio Belmonte. And I did the work in the OnTox project. So I also love help from the OnTox team. So now I'm actually quitting age-based modeling and writing my thesis at this moment. But if you have any questions or want to contact me for other stuff, you can find my personal email address. Are there any questions?
Thank you very much, Jo, for a fantastic talk. Floor is open for question. I have a few, but I'm happy to hear from.
I can ask a question if that's okay.
Please go ahead.
Thanks, Jabez. That was a great talk. I think I might have missed this. I don't see the connection. How did you connect your GRN, like the genes that are causing the problem, and how did they cause the problem? Like, was it linked to some cell division? Like, how did they know not to close in your model?
So I think your question. So
like, is it forces? Did I miss that? I wasn't sure what happened. Like, how did it know that if you remove BMP4, that all of a sudden it didn't close? Like, I didn't know how BMP4 left the.
So basically what I did, I developed this gene regulation network based on biological observations. And all these genes have a specific role, basically. And they induce or they steer cell behavior towards specific behavioral things. For example, BMP4 is very important in specifically signaling in this pathway to cause the cells to bend. So based on adverse outcome pathways, we identified that bending of the tissue is very important to form from a straight line and circle. But if the function of BMP4 is disrupted or disturbed, that can result in cells not getting the proper signaling and thus not acting the proper behavior, which then results in a failure of closure. Does that answer your question?
Yeah, I was missing like the link. So you already assumed that they were going to cause something. Thank you.
Yeah. Any other questions?
I have a couple of questions, if I may.
Yeah, sure.
Yeah, please, Oliver, and then Tom. Yes.
Okay, my first question was, how do you define your different cell types in your model?
How I define those?
Yes.
I define those based on biological observations. So we know that certain cells in certain locations in the neural tube can have specific behaviors. And then in the COMSALT ready, is a cell is similar, then it's like cell type A cell has different properties, can call it cell type B, and so on. And I think there are a total of 20 cell types in the simulation.
They're not defined by their gene expression pattern, are they?
They are both defined, their behavior is defined by gene expression pattern, but for to be able to actually program properly, they also have a separate definition, but their behavior is defined by the gene expression patterning.
Okay. My other question was, how do you define the initial conditions in your model?
I don't define the initial condition, so I only define the initial cell structure.
Number of cells, number of cell types, the cell has to be in a certain state.
Yeah, I based it on the observations of the bud state. I think you can still see my screen, right? So right before this image, there was another image where it's flat. So I based the initial state on these virtual human embryo sections. And that's mainly the number of cells. And the internal gene expression state is automatically resolved by the gene network itself. So there's nothing I need to define. I only need to define that genes are allowed to exist.
Yeah, but the behavior of the network will depend upon the amount of specific gene products, right?
Yeah, not necessarily, because it's relative. All the genes are relative, so no absolute numbers.
Okay.
So if you look at the gene network and you, for example, see, let's see if I have it here. So basically, the network resolves itself at step 1. Certain genes are inhibited by other genes, but if they are inhibited, if they are active, they activate other genes, and you get this automatic patterning across time. Basically, so if I show this state, what you basically see is you have your initial state, but it resolves very quickly into this stable patterning that's comparable to in situ hybridization, the way the gene network is wired. So this is the initial state, and then it evolves over time because some genes either are activated or inhibited based on other genes, which doesn't make it necessary to really initialize a start state, would only allow genes to exist, and then they'll get the signaling if it's their time to be able to active. As long as the biology is properly wired, that's the most important thing. If you don't have the biology properly wired in your model, then I don't think it's possible to achieve this network.
Okay.
And behavior.
If I may, I have one last question regarding the subtypes. How do you quantify them? I mean, how do you decide it's subtype A or subtype B? Do you do that by any additional mechanism or algorithm, or do you do that by eye?
I did by eye. So basically, because you look at sort of as at it like clinically, right? So I defined it by, okay, well, how, if you are a clinician and you look at neural tube defects, how would you clinically quantify, for example, if something is subtype A or subtype B? Of course, something can have multiple subtypes. That's why I call it subtypes, not main defects. So defect can be both disrupted cell delamination as well as asymmetry, for example.
Okay, thanks.
It's really a clinical point of view. So it would be more powerful if you write an actual AI model to do the quantification, but I unfortunately didn't have time to develop one. Although I think the James Glaser group did make one AI model, a UNet, I believe, for agent-based images.
To analyze the images and quantify
Yeah,
cluster
that would
them.
be better, but I did it manually.
Okay, thanks.
Tom.
Hey, Tom here.
Hey.
Very nice, like usual. I have two questions for you. One is relevant to the model and one probably not. So I'll ask the relevant one first. When you do these knockouts or knockdowns, you're starting from your zero time with the introduction of the perturbation, right?
Yeah.
Have you tried trying to pinpoint or localize the critical time period for some of these phenotypes that you're producing? In other words, if you were to introduce your perturbation at a certain time within time window within the model, can you localize what the critical phenomena really is in terms of setting off the abnormal phenotype?
I did indeed start at time zero, so the perturbation is always there. I think you're able to pinpoint some of those. I know that, for example, sonic hedgehog is most essential at the beginning and wind is more essential later, but I did not simulate those scenarios. But I think you can use the age-based model to test those kind of situations.
Yeah. Okay, and the irrelevant question. So I noticed in looking at some of your zebrafish that the swim bladder looked like it was affected as well, which is a feature that's not in human embryos, obviously, unless I am misinterpreting your diagrams or your photos. But if the swim bladder is critical to some aspects of zebrafish development, can you think of ways of potentially introducing those external influences into the model to maybe tease out what might be, say, a species-specific effect, even though we know that the pathways are generally conserved, but the structures are not necessarily conserved?
Yeah, I think because the fish is not a fish, you still have these species differences effect. And indeed, swim bladder is not necessarily related to the neural tube, I suppose. But for example, I think there is, if you look at the fins, for example, the limbus, I think there are comparisons there, although they're different structures with the magnet, how it's formed.
Thank you.
Yeah, thank you.
Lutz.
May I thank you? Your fascinating work. Thank you again for presenting. And I also have two questions. One is regarding your zebrafish work. It seems that your RNA-seq analysis concerns all of the cells of a zebrafish because your 2D images also focus on the whole fish, or do you sort cells from particular portions of the embryo?
So no, it's like the RNA sequencing is the whole fish. So I didn't try to cut up the fish at night. So it will be hell probably. So it's the whole fish. The thing is, if you look at these images under the microscope, the specific time points, like almost the whole fish is the developing neural tube in the earliest stages. Like at 24 hours post fertilization, you see also the eye and the head to be getting actual shape. But in these early stages, most of the fish is the neural tube actually, so we didn't find it necessary to isolate specific cells. Although,
however, your model has like 4 or 5 different cell types.
yes,
and the effect of your perturbation may be very different in the different cell types. As you say, sometimes gene A, whatever, is upregulated, but that may be differing between the cells that you represent in your model. So may not be equivalent to averaging over all the cells and then putting the effect into one particular cell type in your model where this gene plays most of a role.
I think if you isolate the cells, you will see better effects. If you can actually localize the cells, for example, if you can localize the neural crystal specifically and see their pathways, I think you have a stronger effect in the model, because now you're really predicting average cellular behavior, basically what would happen on average. Although the current healthy state model is parameterized in a way that it can act on small genetic differences, so to say.
But it may also be the other way around, that when some other cell type reacts strongly to your perturbation with a gene up-down regulation, and you measure this, but it's not affecting the particular cell types in your 2D neural tube section, then maybe it doesn't play a role, although it plays a role in many other cells of the fish except for the neural tube. So it may work in both ways, that you get different results if you were analyzing the perturbation in a more cell type specific manner around the neural tube, I believe.
Yeah, probably. I think if you do single cell, it's way more accurate than how we did it. Now you really get a general idea of what happens across time, but not necessarily what does every cell do. If you actually have this single cell data, which there is online, I think, then you can see much better results and also make your model behavior better or improve it in a way. It would be a nice follow-up project actually to see if people manage it. So all the data, all the modeling work will be on GitHub, open source. So people can continue working on this model, try to maybe also integrate single cell data. I think it will be a massive improvement over how we did it now.
And I think your work is perfect motivation for this next step, because you show how sensitive the process is to these changes. So I think this is required, but then one also probably needs to go the second step. Now, a forward-looking question. There is this 3 in CompuCell 3D, right? So what would change in your model if you included this long axis of the embryo as it is currently done? I think you would have the defect all along the axis, but in your motivating slide with the embryo, you had a very small localized defect down the back where you have this open back problem. What would you need in addition to have this defect not symmetric or uniform along the whole axis, but have this localized neural closure defect at the very low back?
So what we know of like the whole axis of the neural tube is that there are slightly different processes responsible for the closure at each, let's say I-axis place. So like if you have completely open, that's like the most severe case. What you more often see is that there are small parts of the neural tube open. So if I would go to 3D and I would do it correctly, right, then are fused, but other places not, and you see like a mix of open and closed across the spine, basically.
But do you know how, for instance, the temporal aspect, as you saw with the somatogenesis, that basically gives a sequential processing along the axis, how that may factor into this defect first not occurring and then not occurring later? So that it is limited to some specific position?
I think on the rostral-caudal axis, there are indeed like for example, there are different protein gradients higher at the most left end and low at the other place, and the other way around. So what you really see is maybe that at some places the timing is misaligned, where it leaves it open, but in other places the timing is good enough still. And what you also really miss in these 2D simulations is, for example, the physical forces that are generated by bending of the neural plate. So you can imagine if you have a longer tube and the most front part starts to tuck it closed, then the latter part will also follow automatically because these physical forces are generated. And these are also essential informations. Also, what's also kind of interesting, like I did somite development in there in 2D, but somites are actually round. So it's like a flat slice of somite. But if you look at the actual somites, there will be circles. So there's also one essential 3D aspect that's currently missing. So in some simulations, in my case, you see like ECM trapped in the somite, but of course, normally then be able to diffuse from the other side out of there, what's not yet fused in a way. So that are all these small elements that are omitted in these 2D situation, but are definitely present and important in the 3D situation.
And great work already in 2D. Thank you.
Thank you.
Job, you need to plan another year or two for your PhD work. There's too many interesting things to do left.
Yeah, maybe after I'm done with my postdoc, I can come back and then finish what I started.
Nice. Olivier, did you have another question or just left?
More of a comment than a questions. Actually, what you want as data to confront your model to is spatial time-dependent transcriptomics. Then you would have everything to fit the model. Because the model is at the single cell and as spatial information. So you want to have spatial transcriptomics information regarding at different steps during the development. That would be 4D spatial transcriptomics. That would be just the perfect type of data you would need to really fit the model.
Yeah, that would be the best. We can write a proposal and then do the experiments and then try to integrate it in the computational model. Then we have like a lot of stuff to do in the coming 3 years.
As the previous colleague said, it would be a good motivation to do those experiments because the model would be there to just make sense of it.
Yeah, thanks.
Well, if there are not other questions, we are on top of the hour. I will have just a last one that relates to the price of most reproducible model, and I will steal your share view while you answer the question. What can you say a few words about reproducibility in this kind of model? So how this is very nice and very complex as well. How do we make sure that eventually someone will just build on top of that and won't need to redo something else, etc.? Or how can we at least make sure that everyone can reproduce the results and use them?
Well, I think reproducibility is of course very important. So at least what I am doing for it is I put everything open, accessible on the GitHub, as well as all the ways I did analysis. So the full RNA sequencing analysis pipeline will also be there, as well as the integration, how to integrate the genes in the computational model with all the relevant references. And I tested in a new environment, right? Of course, you need consult for these and need to have the same version, or maybe I think it's exclusive. So you can also run my 4.4.1 version at 4.7. So you can just download that and then you run straight from the command line. Do the parameter sweeps, so everything will be accessible, and then it should be as reproducible as possible, I suppose.
That's super, and this is very important as well. Well, on these notes, I think we can close. I will just say a few words. Well, first of all, I want to wish everybody a happy Christmas break, festive season. Please check out our channel, Immobile here, but also our usuals, Slack, AmagMSM, YouTube, and GLIMPRINT. And with that, again, happy festive season to all, and have a good day and afternoon to you all. Thank you very much for joining us today.